Okay, before we lock in, there's some super important vocab that we need to know. We're going to start with sarcolemma. This is the cell membrane, because as you know, here's a cell and has a membrane. And then next, we have the sarcoplasm. That, believe it or not, is a cytoplasm. It can get inside of a cell. So lemma and plasm. Then we have the sarcoplasmic reticulum. Boom. This is the calcium reservoir. So there's calcium. You remember what that is. All that is stored in this bad boy. Next, we have T-tubules. T stands for tunnel. These are tunnels that go through the sarcolemma. So remember the sarcolemma, there's a membrane. Bam, little T-tubule. Stuff can go through that. It's pretty awesome. Next, we have a thick filament. Thick filaments are made of myosin. It's a type of protein. And they're kind of shaped like that. They have a golf club looking head. That's important to know later. Then next, we have thin filaments. Because where there's thick, there's got to be thin, obviously. And the thin filaments, they're made of actin. That's another type of protein. I remember that actin is a thin filament because it sounds like thin. That's what Mr. Thurman told us. We'll see if that helps. Next, we have tropomyosin. My hand is really dry. Ew. That blocks the active sites on actin. As you know, there's actin. It has an active site for stuff to bind to. Then there's tropomyosin. This gets in the way. And then next, we have troponin. That's a calcium binding protein. It's connected to tropomyosin. There it is, that little handsome fella. So the calcium, remember the calcium comes from the sarcoplasmic reticulum. That stuff comes out, and then when it's necessary, it binds to troponin, which is on the tropomyosin. Next, we have titin, like the big boy from Megamind, the redhead. Titin is a springy protein, like remember what a spring is? Boing. That stabilizes thick filaments, the ones made of myosin. Because we use them a lot, but we don't want them to overstretch, so titin stabilizes those. That's important to note. It's the biggest protein in the body. I'm going to circle it, because I put it on your Quizlet too. Next, we have synapse. You know what a synapse is, because we talk about neural synapses a lot. It's the point where a nerve meets its target cell. In the case of muscle physiology, this is called neuromuscular junction. Bam. That's when the nerve's target cell is a muscle fiber, so it'll meet this thingy. Next, we have synaptic knob. Synaptic knob is the end of a neuron, this little knob like a door. This releases acetylcholine, ACH, and this is the neurotransmitter, and that's really important to know later. And then the synaptic cleft. This is the gap between the synaptic knob at the end of the neuron and the sarcolemma of the muscle. There's this goofy little gap here, and that's called synaptic cleft. Next, we have depolarization. We have this muscle here. Depolarization is when sodium Na+ goes in here. And then repolarization happens right afterward. This is when potassium gets kicked out, K+. Smell you later. And this quick voltage change that is caused by this creates an action potential. This action potential only happens in active cells, not resting ones. It only happens here, like when we use our muscles a lot, basically. Now we're ready for the big juicy part of muscle physiology. Our first phase, because there's 4 of them, it's called excitation. In this, there's a neuron, and then there's a synaptic knob. The synaptic knob has calcium channels in it. The neuron opens the calcium channels in its knob, and then the calcium stimulates the release of acetylcholine. Remember that? The neurotransmitter. So we have the neuron and the calcium. Bam, bam. Spits out acetylcholine. So like calcium basically the middleman here. And acetylcholine goes into the synaptic cleft. And then it meets the sarcolemma. And then that allows for the voltage change to happen. Sodium goes in, and then potassium comes out. When the sodium goes in, that's depolarization. When the potassium comes out, that's repolarization. Remember, this quick voltage change creates an action potential. Next, we have our next phase. It's called excitation-contraction coupling. So when you have this action potential, and that goes down the T-tubules in the sarcolemma. Remember the T-tubule? That allows calcium to come in. The calcium binds to troponin on tropomyosin. So here we have tropomyosin. He's the big guy because he holds all the troponin. Here it is in his hand, like a burger. Tropomyosin. And that is troponin. Well, remember the action potential allowed the calcium to come in for the T-tubule. That calcium binds to the troponin. Boom. They're kissing, basically. And then that tropomyosin is now able to move. He gets out of the way, this big fat guy. And now we have an actin filament. This fin filament, there's an active site. Now that the fat guy is finally out of the way, the active site on the actin is exposed. So, yeah, the action potential allows calcium to go in, it binds to troponin, and then tropomyosin moves out of the way and exposes the active site of the actin. Next, we have contraction. Here is myosin. This is the thick filament, remember? And then there's an enzyme called myosin ATPase. That phosphorylates basically, it activates something in the myosin head. And then the myosin head is able to cock like a gun. So it was, I guess it's supposed to be straight. And now it moves this way. And then, now that the myosin head has been activated, it can bind to the active site on actin, which has been exposed because remember tropomyosin moved out of the way. Got his big fat butt out of here. And that thing is active now. Now that the myosin and the actin have finally bonded, they're like friends now, it made the cross bridge. That's important to know. Now the cross bridge is here, then there's a power stroke. Boom. You see that big stroke? But then a new ATP comes in, and then that breaks the cross bridge. His heart shattered. Boom. Tragic. Next, we have relaxation. We're back to the nerve here, the neuron. This neuron tells the ACH to shut up. So this one tells stop secreting because it doesn't want it to come out. And then we have acetylcholine esterase. Acetylcholine esterase is an enzyme that breaks down acetylcholine and that sends it back to the synaptic knob. Because remember, that's where it came from. Because we need acetylcholine to go back in the synaptic knob, because if it doesn't go back in, it just keeps contracting the muscles, and that causes spastic paralysis, like tetanus or rigor mortis. And then, now that the ACHE has broken down the ACH, the calcium gets stolen from tropomyosin. Remember the big boy? That calcium, it was binding to the troponin, but now the calcium is gone. It goes back to the sarcoplasmic reticulum, because that's a calcium reservoir. Now tropomyosin is sad, so it moves back to where he came from. Then he blocks the active site of the actin. So the myosin can't bond to make another cross bridge again unless we need it to. So that's basically it in this relaxation. The ACHE breaks down the ACH, then the calcium leaves, goes back to the sarcoplasmic reticulum. Then tropomyosin blocks the active site of the actin again. There we go. It's a lot, but good luck.